So I'm back after the battery recharge, which in real time is probably closer to 36 hours, but I'm back. So I believe the last thing we were talking about was the relative abundance of specific antibody in the blood. And we're looking at weeks after first exposure to antigen A. So there is some antigen that's been picked up somehow, either processed through phagocytic cells or picked up by helper cells here, but this T helper cell has got this antigen. It wanders through the lymphatic system, finally finds a B cell that will give it the time of day, and this B cell recognizes that antigen because of its ability to fit into the B cell receptor. That signal transduction process triggers differentiation through mitosis of plasma cells. These plasma cells then begin to secrete antibody. This antibody that's showing up a little less than a week after exposure is the amount of time it takes this whole process to get cranked up. Now we've got antibody being produced a little bit before a week, and the antibody that's produced in response to that antigen begins to show up in large amounts in the serum. Okay? Then as the antigen is cleared from the body, and keep in mind this little antigen may actually be a little part of a surface protein on a bacteria or a virus, so we're clearing out these cells here as we design essentially these antibodies to fit these antigens on the surface of proteins, on the surface of bacteria, viruses, and other pathogens. Okay? The immune response then begins to taper off as the body clears itself of these antigens. So this is this clearing out. Notice that even after 4 weeks after exposure to the antigen, the body is still producing a low level of these antibodies. Okay? Now, this is referred to as the primary immune response because it's the first one. This is the first time the body has seen this bacteria and picked up that antigen to stimulate the immune response, which was ultimately the production of antibodies against these antigens. Okay? Now, some point in the future, let's say that this same pathogen shows up. Well, in this case, the immune response kicks in much quicker. Notice that it doesn't take almost a week for production of antibodies. You have production of antibodies almost immediately, and the size of the response in terms of the relative amount of these specific antibodies in the serum is much larger under the secondary response. And they even show us a primary response to antigen B so that we can see how the primary response to an antigen differs from the secondary response to an antigen. Okay? And the reason that we're able to crank up production so fast is that because of this primary response, you now have an army of these memory cells, right? Because after initial stimulation, you produced a bunch of these memory cells, and these memory cells are sitting around, and when a T cell comes in contact with one of these memory cells, it's accessing a much larger army of initial cells that lead to a much larger army of plasma cells and associated secreted antibodies. Okay? This secondary immune response is characteristic of acquired immunity, the idea of being able to recognize something that you've seen before in the distant past. Now, we've taken advantage of this idea of acquired immunity through things like immunizations. Right? In immunizations, what you're doing is basically giving someone the antigen to trigger the production of a primary response, so that in the future, when they get the pathogen for real, they're already into the secondary immune response, which is a much faster and much more intense response to that antigen presentation. Now, the interesting thing with the immunizations is sometimes these immunizations are live immunizations. Often what's been given to individuals are attenuated, which means if it's a virus or a bacteria, in the case of a bacteria, it may be alive, but you stress the heck out of them so that they really can't cause any problems. You attenuate them. In the case of a virus, viruses are dead, but you could attenuate the viruses, meaning that the virus has been damaged. It's not capable of doing what it's supposed to be able to do because you kept it at a different temperature, so that crucial binding sites were a little bit different shapes. But the main site that provides the antigen for immune system stimulation is still present to trigger that primary response. Right? Some immunizations, what are given are dead cells, and in some cases you give just the pieces, the little antigens that trigger the immune response. Now, I don't know enough about immunology to be able to tell you why it is that some vaccines have to be given as attenuated and some vaccines can be given as the antigens themselves. It seems to me right now, at least, to be specific to the type of pathogen that you're dealing with. Okay? All right, so the idea of giving someone the primary response so that in the future they're capable of mounting a much stronger secondary response. Okay? In some cases, you haven't been immunized to a particular disease, but there is no quote-unquote cure for that disease. So one of these might be rabies, right? The pathogen that causes rabies. You can get immunizations for it. There are a series of immunizations. But if someone gets bitten by a rabid animal, giving them the immunization at that point isn't doing anything beyond what is already happening in the body. Because if it's the first time they've seen the rabies pathogen, they're mounting an immune response to it. Giving them a vaccine at that point, after they've already been exposed to the antigen, isn't going to do anything extra. So in that case, what's given are antibodies. Right? You get rabies immunoglobulins that allow for the antibodies to already be present to allow for a more efficient clearing of the pathogen. You're just amping up the number of antibodies in their system so that the number of antibodies mimics a secondary response. Okay? All right. Now, once you've got the antibodies that have been produced, right? Here's the antibody itself, this little site here, and here these are the antigen binding sites. There are the antibodies bound to a particular antigen. Now, when we think antigen, we think anything that triggers an immune response. But if this is the little peptide that's been picked up by a T cell and presented to a B cell, it's possible for some B cells to recognize this portion of it, because that's their B cell receptor, others to recognize that part, and maybe another B cell with a third shaped B cell receptor recognizes a different part. So my point is, you can have multiple B cells that have gone into differentiation into plasma cells and memory cells. These plasma cells are producing antibodies that recognize different parts of the same antigen. So that's what they're showing here. there's the antigen that triggers the immune response, but there are currently 3 different antibody types that recognize the same antigen by binding to different parts of that antigen. Right? It's just how the T cell got a hold of it and presented it. Now, once these, sorry, let me show you this here. Once these antibodies are released into the serum, they begin to bind whenever they bump into the antigen. It's just in this case, the antigen, instead of being some free molecule, is currently bound to a protein on the surface of a pathogen. So the antibodies are binding to the surface, and now you've got the pathogen with antibodies stuck all over it. Now these antibodies are marking that particular pathogen for destruction. Okay, and there are a couple of different mechanisms for destruction here. The first mechanism is you've painted this bacteria or other pathogen with the antibodies, and then you simply need other immune system cells to come along and clear out anything that an antibody is stuck to. And the cells that do this, a lot of them are the macrophages capable of phagocytosis. They will do phagocytosis with anything that has antibodies stuck to it. So here, the binding of the antibodies to the antigens on the surface promotes phagocytosis. So you've got the antibodies painting the bacteria for destruction. The macrophages recognize these painted cells and then destroy them. Okay? All right, the second way that this can work is non-cellularly. This is basically a cellular pathway to destroy the pathogen. The other mechanism is more of a non-cellular process. This non-cellular process involves a group of proteins in what's called the complement system. A group of immune proteins. Okay, and these proteins are found within the serum and the lymphatic system. And where you've got antibodies painted onto the surface by binding to antigens on the surface of some cell, these complement proteins begin to self-assemble around these antibodies, and then the proteins then produce pores on the surface of membranes, and once this pore formation is complete, the cell is going to die because it can't regulate what goes in and out anymore. Okay? So an antibody stuck to the surface either results in destruction by the cellular component of the immune system or a non-cellular component called the complement system. And then the last thing to do here is there are different types of antibodies that are found within us. All antibodies are not created the same. So I just wanted to show you a few of these 5 classes of antibodies. The most common one here is this IgG. This stands for Immunoglobulin G. And typically when people draw antibodies, they're talking about IgG, this very simple one here. This particular one is very good at promoting cellular destruction of whatever the antibody is bound to. It's less effective in activating the non-cellular component, the complement system. Okay? And then here, only the IgG crosses the placenta and confers what's called passive immunity onto the fetus. Okay, so if there were an infection and mom recognizes the infection, she can then give the fetus the ability to fight off that infection by giving the fetus the antibodies that are required. Okay? All right. Here, IgM, Immunoglobulin M. This is a different looking one. It doesn't have like a single antibody. It's made up of a pentamer. It's 5 separate antibodies that are connected together like this. And what this does is bring together a bunch of different pathogens that have the same antigen. So if this one is stuck to that one, here I've got 5 bacteria all joined together. That makes the process of phagocytosis a little bit more efficient. Okay? This is actually the first one that's produced in response to an infection. Okay? All right, the next one, IgA, right? This one is basically a dimer. Right? The antibody, the immunoglobulin is 2 antibodies stuck together. This one is found in all sorts of secretions, tears, saliva, mucus, and breast milk. This one would then be passed on to the infant during nursing to provide the infant with passive immunity. If mom has been sick with a cold, her antibodies can then confer immunity to the infant. Okay? All right, the next one is IgE. Okay? This one is very much responsible for allergic responses, that is inflammatory responses. Okay, it hypes up histamine release from the basophils and the mast cells that we talked about during the inflammatory response. And then finally, IgD. This is the one that's primarily on the surface of the B cells that haven't been exposed to antigens. Okay, so the T cell, when it presents to a B cell receptor, that's this one. Okay, this is the one that stimulates the release of the other ones. Okay. All right, so I think we just have one kind of partial class left in AFF this semester. I'm basically done with the material that we need to cover. I feel